This is a revision video on cell fractionation. There are 3 questions to have a go at. So pause the video, attempt those questions, and then you can play the video again to check your answers. Okay, so let's go through the answers. Question 1, what is the purpose of cell fractionation? Why would you carry out this procedure? Well, you may have tissue from an animal or plant, and what you want to do is to isolate the organelles. That's the point of this process. It's to isolate the organelles. That's really important to remember throughout this process. It's about getting the organelles. Okay, so question 2, let's move on. So there are 3 steps to the cell fractionation process that you need to know. You also need to know about the solution that you use to carry out this procedure in, but we will cover all those things. So let's imagine we have a tube, we have our biopsy in that tube. The first thing we would do is we would carry out homogenization. And you must describe that process. It's not just writing homogenization, you need to describe what you mean by that. And that is the breaking up of tissue and the breaking open of cells by mechanical force. And the whole point of that is to release the organelles. So really important you remember that. We are breaking open the cells. Don't make the mistake of thinking that we are protecting the cells here. We actually don't care about protecting the cells. We want to break open those cell surface membranes to release the organelles into solution. And so you can use a specialized blender or a specialized device called homogenizer. So here it might be a special device that fits very tightly into the tube. And so when you twist that around, it creates this mechanical force, and that causes the tissue to break apart and then causes the cells to break open as well. So as part of this process, you also need to be able to state features of the solution that you use for this homogenization process. And this is all about protecting the organelles. So you need to state that the solution is cold to reduce enzyme activity. And so when you break open the tissue and the cells, you're going to release some enzymes. You might break open some lysosomes, for instance, and they will release enzymes. You don't want those enzymes to be damaging the organelles. You want to study the organelles at the very end of this, so you need to protect them. So that solution needs to be cold to reduce enzyme activity. The solution needs to be isotonic. That is, it needs to be an equivalent water potential to your organelles, because you do not want your organelles to gain or lose water, which would cause damage. So isotonic to prevent organelles being damaged by gain or loss of water by osmosis. And finally, that solution needs to be pH buffered. It needs to be maintained at a fixed pH. And that's to prevent denaturing of enzymes in the organelles that are important for the organelle function. Remember, at the end of this, you would want to study those organelles. So you want to keep them intact, you want to keep them working. You don't want to damage the enzymes that are working in those organelles. So it needs to be pH buffered as well. So you can't just list those features, you can't just write cold, isotonic, pH buffered. You must say why it's cold to reduce enzyme activity. You must say why it's isotonic to prevent organelles being damaged by gain or loss of water by osmosis. You must say why it's pH buffered to prevent denaturing of enzymes. Okay, so you need all those bits of information. Okay, so that's the first step. Let's move on to the second step. That is filtration. So our homogenization process is not going to be perfect. We're going to still have some bits of tissue, maybe unbroken cells. We need to get rid of those. So we pass our homogenate, that solution that we've carried out homogenization in, that will have organelles but also other bits in there, through a filter. So if I have very small holes in it, and that will catch unbroken tissue and cells. And so our homogenate should then only contain our organelles. So that is step 2. Okay, let's move on to step 3 in the cell fractionation process. And so step 3 is ultracentrifugation or centrifugation. And what does this involve? As described here, we use a device called a centrifuge that allows us to put tubes in that contain our homogenate, and we can spin those at a fixed speed, and each time we would then increase the speed, and that will allow us to separate our organelles by size and density. This is a very simplified diagram. If you just imagine a rotor, that's part of the centrifuge machine, and that will spin our tubes. And when we spin those, we will get a pellet at the bottom of the tube. That will contain the organelle that's moved to the bottom of the tube. And then in our supernatant, that is the solution at the top, we would have some of the lighter organelles. And each time we spin the tube faster and faster, we end up getting the lighter organelles. So we start by pelleting the larger or heavier organelles, and then we would take the supernatant, spin that again, and then the next lightest organelle will be in the pellet, and then we can take the supernatant again and repeat that process, increasing the speed of the centrifuge each time to get the lighter organelles. So let's take a look finally at question 3. So I ask you to match the following organelles with the correct pellet: chloroplast, nuclei, mitochondria. So we would have our homogenate in a tube. We would begin by centrifuging at a low speed. So our first centrifugation would be a relatively low speed, and we would get a pellet, and we would have the heaviest organelle in that pellet, and that would be the nuclei. So I'll reveal that in a minute. We would then take the supernatant and we would put that into a new tube and centrifuge that again at a faster speed. That would pellet the next heaviest organelle. We'd get that in the pellet. That will be our chloroplast, if it is a plant cell, of course. We would then take the supernatant of that, we would put that into a new tube and centrifuge that again at an even faster speed. That would get our next organelle. That would be the mitochondria. And then we could actually do that again. We could take the supernatant and we could repeat and repeat. And so what we'd get is, as I said, nuclei in pellet 1, that are the heaviest organelles. We get the chloroplasts, so they're lighter than the nuclei. So we'd get those in the second pellet. Third pellet, we would get mitochondria. So the mitochondria are lighter than chloroplasts and nuclei, so we'd require 3 spins. If this was an animal cell, if we wanted to get the mitochondria, we would just require 2 spins, and the mitochondria would be in the second pellet. It's worth mentioning that ribosomes would end up, they are the lightest organelle or smallest organelle, so we would need to spin at a very high speed for those. They're the lightest. Something that you don't need to know about, but maybe worth being aware of, is the other organelles as part of this homogenization process will get broken up, and so they will form what are called microsomes. So we can't really say that we would spin to get the rough endoplasmic reticulum endoplasmic reticulum, Golgi apparatus, because during that homogenization process, they would be broken up and they would form these sort of lipid droplets called microsomes. But you don't need to be aware of that. Okay, great. That is cell fractionation. So you need to know all those details. So repeat this as many times as you need to get all of this correct, and then you can move on to trying exam questions on this topic.